The name of our presentation is Forensic Rape Analysis, and I'm Ifeoma Nwaedozie. And I'm going to interview our What is rape? Rape is forced, unwanted sexual intercourse and can sometimes be called sexual assault. 911 Rape is an informational service provided by a Rape Treatment Center at Santa Monica, UCLA Medical Center. 911 Rape provides support for sexual assault victims.
Okay, introduction. DNA evidence is the key to solving crimes in a scientific manner. DNA is an acronym for deoxyribonucleic acid, which is made up of 4 bases: guanine, cytosine, thymine, and adenine. Trace evidence is evidence that includes hair, fibers, paints, explosives, glass, headlamp filaments, or anything that can be found left at a crime.
What evidence is left behind? Rapists leave behind one main type of evidence, DNA. It can differ depending on the circumstance and situation.
Hair analysis. The examiner compares a variety of factors of hair, including color, coarseness, granule distribution, hair diameter, and the presence or absence of the medulla. Hairs grow out of tiny pockets of skin called follicles, and the part of hair that attach to the follicle is called the root hair. Each strand of hair has 3 layers: cuticle, cortex, and medulla.
Hair analysis methodology. Obtain the hair, examine it under a microscope, check for the medulla, cuticle, scales, and distribution of pigment granules. Make drawings and record observations, and determine if the hair is human or animal. In humans, the medulla is usually thinner, and in animals, thicker.
This is a picture that shows the difference between an animal and human hair. In this picture, you can see the medulla is obviously thicker as an animal. With the human hair, it's thinner than the medulla.
Here's another picture, a deeper picture of the hair. An innermost layer of the medulla, which is an innermost layer of the medulla, which is only presented in the large thick layers. The middle layer, known as the cortex. The cortex provides strength in both the color and the texture of the hair. And number 3, the outermost layer is known as the cuticle. The cuticle is thin and colorless and serves as a protector of the cortex.
Okay, fiber analysis. Fiber is the second most common type of trace evidence that is left behind. It can be found as an individual fiber, threads, or fabric. The 2 categories of fiber include natural and animal fiber, natural and artificial fiber. Natural fibers are made up of mineral, vegetable, and animal. An artificial fiber can be regenerated vegetable fiber that has been changed, such as the addition of acetate.
Here's a fiber solubility chart. I'm just going to use my mouse. First, you start off with an unknown fiber. If you want to find out what kind of fiber it is, you start off with an unknown fiber and you add formic acid to it. If it's soluble in formic acid, it can be an acetate or nylon. So to find out which one of them it is, you add dimethyl formaldehyde. And if it's soluble in that, it's an acetate. If it's insoluble, it's nylon. If it was insoluble in formic acid, you go on to add nitric acid. It can either be an acrylic if it's insoluble in formic acid, but if it's slightly soluble, it can be silk or wool. To find out which one it is, you add 5% sodium hypochlorite. If it's soluble in that, it can still be silk or wool. So to find that out, you add 75% sulfuric acid. If it's soluble in 75% sulfuric acid, it's silk, and if it's insoluble, it's wool. If it was insoluble in nitric acid, you add hydrochloric acid. And if it's soluble in hydrochloric acid, it's rayon. And if it's insoluble in hydrochloric acid, you go on to add 75% sulfuric acid, which can make it either cotton or linen. Dimethylformaldehyde is used to identify polyester or molded acrylic with the addition of acetone.
DNA analysis. During a rape, there are many forms of DNA transfer evidence left behind. This includes blood, other bodily fluids, skin cells, and skin cells. This type of evidence provides DNA which is replicated with the use of polymerase chain reaction and analyzed using gel electrophoresis.
Okay, here's a chain, here's a chart of polymerase chain reaction. The first step, the DNA is denatured and primers are attached to each strand. A new DNA strand is synthesized behind primers on each template strand. Step 2, 3, and 4 are all the same, and step 5 is continued rounds of amplification to produce large numbers of identical fragments. Each fragment contains the DNA region of interest.
Gel electrophoresis. The replicated DNA is then put through a gel electrophoresis process. Electrophoresis is a technique used to separate charged molecules. DNA is a negatively charged molecule, so this is subjected to an electric current through a matrix of agar gel.
And we have a video.
Because proteins and nucleic acids are large molecules with many charged chemical groups, each molecule has a different specific net charge. This characteristic can be used to separate them. The apparatus used to do this is made of sheets of plastic or glass with a space in the middle. The gel matrix is cast in the space such that there are a number of wells for samples at the top. At the bottom is a reservoir for buffer and a positive electrode. At the top is another reservoir and a negative electrode. Samples for analysis are added to the wells at the top. Usually, one of these is a mixture of molecules of known size called standards, against which the unknown samples can be compared. Sample molecules which are negatively charged move toward the positive electrode at the bottom. Those which are the most negative move the fastest, and thus separate from the others. In reality, you can't see the samples as they move through the gel. When the electrophoresis is complete, the gel is stained to reveal the proteins.
Gel electrophoresis continues. After the process of gel electrophoresis is completed, the DNA is compared with the DNA from suspected rapists in the combined DNA index system, or CODIS. This makes identifying suspects easier.
Forensic rape analysis conclusion. In conclusion, rape is a violent crime that occurs to about 1 in 4 women and about 1 in 6 men. There are many types of evidence left over from a rape crime scene. This is why it's easier to find a culprit.
Future works. In the future, we would like to actually work with the DNA taken from a rape crime. We will use this DNA to help identify the culprit and successfully solve the crime.
These are our references.
I'm Melina.
First Community College staff.
Dr. Stat. We'd like to acknowledge Dr. Thomas Brennan and Dr. John Molina.
Bronx Community College and staff.
Dr. Stat.
Harlem Society.
And our audience.